This webinar will talk about Environmental Scanning Electron Microscope, or ESEM, at Hamline University. Includes 3 parts: detectors, specifications, and applications. Josh and Maddie will demonstrate how to use ESEM for our research project. They will talk about enhanced student education and research experience. First, we wish to thank our generous donors and Hamline University for the acquisition and installation of the ESEM. The Hamline community will have free access to the ESEM for education, research, and service. This is a photo of Zeiss EVO 15 ESEM. Mainly include 3 parts. Here is the electron source, and column include electromagnetic lens, and here is the sample chamber. When the electrons arrive on the surface of samples, can have different interactions, generate different signals, and provide different informations. For example, secondary electrons can give information on topography. Backscattered electrons can generate the contrast or atomic number contrast image. And characteristic X-ray can provide information on composition to use elemental analysis. The ESEM at Hamline has 5 detectors, include secondary electron detector, backscattered electrons, NJ-dispersive X-rays, scanning transmission electron microscopy detector, and also has another detector used in the variable pressure or environmental mode. This is specification: acceleration voltage from 200 volts to 30 kV, operation pressure from high vacuum to 3000 Pascal. Resolution is 0.9 nanometer at 30 kV for secondary electron detector in vacuum. At 3 kV, resolution is 2.8 nanometers. So what is nanometer? For example, is human hair the width around tens of micrometers, or 1 micrometer equals 1000 nanometers. So that means definitely use SEM, you can see a lot object invisible to our eyes. And this is a photo to show the chamber, how it looks like. And this is the sample stage, can move along X, Y, Z, and also can rotate and tilting the samples. We can have the different method to attach the sample to this sample stage. For example, this one is a stub, so that means we use 2-side conducting copper tape. So one side use adhesive attached to this sample holder, and another side we put sample on this surface. And the sample size can be 250 millimeters, and the height is 145 millimeter. You see how small you can do it. So definitely you also, for example, you can disperse some powders on this conducting tape. You can observe it. And this is the chamber dimension. So I will talk about how this is a different acceleration voltage and operation pressure to affect the imaging. This one is an image of light microscope of E. coli. For optical instrumentation, the resolution is around half the wavelength. The wavelength of visible light is ranged from 400 nanometer to 700 nanometers. And how to improve the resolution? So that means we need to use shorter wavelengths of electromagnetic waves. So that means we will consider electrons can be considered as wave, as much shorter wavelengths. That's why it has much higher resolution. We can observe more detail on the surface of the E. coli. Another example, if a piece of bread was left on the side for a couple weeks, we know that is going to see some mold on the surface. How this mold under electron microscope. This magnification is 625. We can see it looks like a chain of sphere particles. We know the highest magnification of light microscope will be around 1000. So this one image take is 1250 magnification. So we can see it also has some branches and also has particles as a tip. It's based on this observation, which is how this is bread mold under electron microscope or under light microscope. You may describe a lot of spherical particles, and it also looks like the chains. And surface seems like smooth. However, if we further increase the magnification, because the resolution of SEM, we can see a lot of detail on the surface. The surface is not smooth, has a lot of spike on the surface. So that means SEM definitely can provide more detail and significant information. It talks about has a different acceleration potential. So we give example air crystals. We look at it under 20 kV. Then the same area, how does it look like at 1 kV? As compared here, definitely has more detailed information on the surface can be revealed at 1 kV. Because it's at 1 kV, electrons have less energy. So that means penetrate less. So that means can shows more details on these surface features. We can look at another samples for the gold particles at 20 kV. Look at this surface, it's rather smooth because we can see not much feature on this surface. At 5 kV, we can see shows definitely has distinctive feature on this surface. We mentioned about has different detectors. So which different detectors can provide different information. For example, we characterize multi-watt carbon nanotube with platinum nanoparticles under secondary electron mode. So definitely we have like one-dimensional nanostructures. And if we look carefully, we can observe some particles. How does it look like under backscattered electron detector? It's very obvious we can see, and this is one-dimensional structure, not that obvious. But this is nanoparticles stand out, has much better contrast if we compare this one. So that is very obvious, has nanoparticles. We mentioned about its backscattered electrons imaging kind of Z contrast or atomic number contrast, because platinum has much higher Z number, is 78, and carbon is 6. So that means huge difference. So that means when we look at backscattered electrons, can reveal some features we cannot observe under secondary electron detectors. Then the same object, we pick up another area. This is for secondary electron image. Then your question may ask, how do you know this is multi-watt carbon nanotube? So that means we need to use scanning transmission electron detectors to reveal the internal structure of this subject. So under this detector, we can see the internal is hollow. Based on this contrast, we can tell this is a carbon nanotube instead of carbon nanofiber, because fiber will be solid, and the tube seems has hollow internal structures. Then we mentioned about this is environmental SEM. though environmental imaging has a lot of advantages. For example, we can combine a partial cooling stage, so we can introduce the water vapor to observe the crystallization of sodium chloride crystals. And this is just like a sword we put under SEM chamber. Then we can start to introduce the water vapor. So that means inside pressure increase. Then this salt will be dissolved in the water, become solution. Then we can control the pressure. So that means we can pump out this water vapor, and this will be recrystallized. So that is a dynamic process. We can observe a large experiment. For example, if you involved with water, you can study superhydrophobic surface. And for the traditional SEM, early you need to coat the samples. If the sample not conductive, you need coating with, for example, gold or palladium, the metal thin layer on the surface to make it conductive. And the environmental SEM, instead of coating, we introduce water vapor, can neutralize the surface charge. For example, is pollen. For the traditional SEM, you'll go through a critical point dryer, try to maintain the structure of the natural status. But sometimes during this procedure or coating with conductive layer, you may change the surface structure. Under environmental imaging mode, so that means instead of coating or drying the sample, you can observe it using different pressure. So there are a lot of applications of ESEM. For example, you can use microscope to observe or monitor the structure of product during the manufacturing and assembling samples. And here I just give some example. For example, for semiconductor and electronics. And this is backscatter electron imaging to reveal like different composition and different layer. And this one is secondary electron image to review a whisker glow on this device surface. This kind of glow sometimes may cause the short circuit. So that use SEM for the manufacturing, you can to analyze for the sample failure or analyze the reason why the component doesn't work, why it works. Here is just like more samples for the life sciences. That is mildew on the surface of a leaf. And this cross-section of orange. And this all sample without coating, without critical point dryer. So you can observe the surface structure. Here is raw materials. Most is inorganic materials. Because you can, based on backscattered electrons, you can see do they have the different composition or elements. And also we have EDX. So they can tell you which elements do you have in the samples, do the phase mapping. For example, this one is mineral map or sandstone reservoir rock. And also to analyze, for example, the paint or materials used for some artwork in the museum. So you can try to restore artwork sometime because you need to try to use the same materials to restore the artwork. And SEM also can use to analyze gunshot residue, paint, hair. For the forensic, this one example is a mark from a firing pin on a gun casing can be used to help identify the weapon used. And this one is fragments from a catastrophic explosive event can be used to determine its source. And right now is Josh will give a demonstration on secondary electron imaging, and Maddie will give demonstration on energy dispersive X-ray spectroscopy.
Hello, everybody. Thanks for joining us for this demonstration of the scanning electron microscope. My name is Josh Zdarsky. I am a freshman. And yeah, so today we're going to be showing you how to get an image with SEM. So up until this point, we had to pump the vacuum chamber down so that it was at air pressure so we can open it. And once we opened it, we put in the samples here. You can see that we took this image of the sample tray. So we have all of our samples in here. We put them in the chamber and pumped it back up again. So from there, we'll be able to go to sample setup, and we're going to pick the right holder. So we're using the 9-stub carousel, so we're going to click that one. All right. So in here, in sample size, we'll be able to change the height of our sample. But since our samples are pretty much flat, it's acceptable to say that the sample height is 0 millimeters and sample diameter as well as good as is. So now that we've done the sample setup, we're going to hit Start Job. And this can give us different types of samples. So if we're looking at a biological sample, like a leaf or something, maybe butterfly wings, some ceramics, coatings, fibers, polymers, metals. This is what our sample is, but we can also do geological samples, food samples, which I think is really interesting. The food and biological samples both. I'm very curious as to see what those look like. And I think you saw some mold in Li Feng's presentation. But yeah, so we also have powders and semiconductors. So we're going to hit metals here. We're going to click OK. And we are going to now hit Start Imaging. So it's going to run the beam up. It has to like power up, as it were. And once that's going, we'll be able to get an image. All right, so now that we have run up beam, now that we're ready to image, so you see we're right here in the center. So this is a little copper sample. This is not what we're going to look at. So what I would first like to go over is the different methods of navigation here. So you can do that. We have a joystick con right here that you can see up on the screen. It's kind of like inverted, so if you move it to the left, then it'll move to the right. Or if you move it to the right, it'll go to the left. If you pull it down, it'll go up. Push it up, it will go down. So that's one way to navigate. The other way is to double-click on the screen, and it'll track. So here, now we're moving. Another way to do it is to go down here, and then to double-click where you want to go. And this way is really useful because it can get you from here all the way over here quickly and automatically, which has been super useful for us. Additionally to that, you can change the image here from photo, and then you can go do a stage view. Now you see a side view. So right. So if we move this over here, you can also get a top view. That shows you where you are. Also a left in 3D view, but personally, I think photo is the most helpful because you can clearly see your sample, where it is, what's on it, and you just click and drag around. This is the sample we're going to be looking at today. So now that we've gone over some navigation, you can see, this image is not quite in focus. So what we're first going to do is I'm going to pop this into reduced mode. This reduces the scan rate. You see that these lines are going down here a little bit faster than when it's off. So here it's scanning at like this rate, whereas in reduced it's going a little bit faster. And that helps the changes you make to pop up quicker. So I'm going to put this into focus now. You can see that that didn't help it, so we're going to go the other way until we focus. Ah, here we go. We like that one. So what we can also do is we can increase the magnification, and we can just focus in on a spot. Why not right there? Ooh, no, just kidding. I like this spot a lot. So let's zoom into that one. Then we can take this to, say, 1K magnification, using this bar on the left. We can also take that to now 2K. About 5K. Very nice. So now we're going to try and get this into focus. Alright. We can also, using the knobs on the top right here, we can fiddle with the brightness, the contrast. I'm going to turn the contrast down a little, focus a little bit, a little too far, so we'll go back. Alright. So that is... Alright, now that we've gone over some navigation and some of the focusing magnification features, let's go ahead and let's find a spot to image. So what we're looking for, we're working on a project where we're aiming to improve the sensitivity of lead detection in drinking water. So these are gold chips that have been coated with biolinker molecule, lead, and then topped with gold nanoparticles. So what we're looking for are gold nanoparticle clusters with some sort of organic molecule. We put this chip under UV light to sort of clean off contaminants. So now we're going to go ahead and zoom in. We're going to look for some spots. And as you see, as we're zooming in here, we've got some interesting points right there. What we're looking for, yeah. This right here kind of looks like what we could be looking for. So I'm going to go ahead and now I'm going to zoom this into, oh, whoa, what does this look like? I guess I'm going to zoom it out a little bit. And we'll find a spot to focus in on. So I'm going to focus in, yeah, we're going to focus in right here. Go through all of the same stuff that we talked about before. Reduced. Now we're going to, oh, look at those just popping out of nowhere. That's interesting. Alright, get this in as much focus as we can. Turn up the contrast, turn the brightness a little bit. There we go. Alright. So, I'm going to go back up to 10K then. Actually, normally it'll be better to do it the focusing in at high magnifications. So that way when you come back out again and then zoom out, it stays in focus. Alright, so now we'll go back up to 5. See, nice focus. We go back out to 2. Again, great focus. Things are looking good. 1K, and you're getting a better sense of the whole region here. This all looks like it could be like a really good spot, potentially. 100, there's that whole region there. Now we're going to go back up to 5 again. We're gonna, now, before we take the image, though, I just want to point out this speed bar. This controls the scan rate. When you click a lower number, it increases the scan rate but decreases the resolution. So inversely, if you increase it, you decrease the scan rate but you increase the resolution. You see the line here scanning down at a lower rate, but this looks oh so much clearer, and now you can really see what I'm saying here when it kind of looks like skin. If you like just take a close look on, say, you know, the back of your hand, all the little cracks and grooves in there, it very much looks like skin. So now that we have our image the way we want it, go back there. So I'm going to hit save. And now it's going to automatically render at a set speed, scan rate. What you don't want to do is you don't want to touch this while it's rendering, because then you'll get some weird lines and streaks that'll kind of ruin your image. You don't want that. So we're just gonna let it go. And now it's named there, demo. You can change the name if you want. We're gonna make sure that it's saved in the right folder here. So if we're gonna go into there, we have a folder named after my esteemed colleague. And okay, so this is where we want it. So we'll hit done. All right, now we have that. Let's save. We'll run it over to Maddie to do some analysis.
Hello everyone, my name is Maddie Sawinski and I am a sophomore applied physics major. And I'm going to be showing you another feature of the SEM, which is elemental composition analysis using X-ray energy dispersive spectroscopy. And this is a really long name, so we just refer to this as EDS. And to do this, we're going to start by first capturing an image of our sample. So we're going to come up here, and this will load up the image that Josh took previously and upload it into this program to use. And using this, there are a few different ways we can analyze our sample depending on what we're looking for. First, we're going to look at point analysis. Using this, we can see the elements that are found in a small point area. And this is really useful when we want to quickly identify an element to see if we are on the right track. So to do this, we are going to come up to Spectrum, and then we can click anywhere in our sample here to get a reading. So I will click right here, and then we can hit Collect. And from here, as you can see, sometimes it takes a moment to get a reading, but then we can look at our pie chart down here, and then as well the peaks here to see what we're seeing in this sample. So we're seeing some interesting things here. So once again, this is from the same project that Josh was talking about for detecting lead in drinking water. And as you can see here, in this particular point, we have some lead and some sulfur, and it also looks like some yttrium, which is very interesting. I'm not sure where that's coming from, but that's part of the discovery process. So we'll wait for this to finish up, and then we can look at our line scan analysis. So next we are going to look at the line scan analysis. So this is similar to the point analysis, except now instead of a single point, we're going to pick 2 points and get a spectrum across a line of the sample. So we don't use this too much in our project, but this can be really useful when looking at cross sections. So to do this, we're going to click up here on line, and we can get a cross section. We'll go across this little cluster right here. So we have a point, and we're going to drag it across and to there, and then we will hit collect, and we confirm. And you can see a periodic table will pop up with some of the elements that are appearing, so then we can hit OK. And as you can see here, this creates a graph of certain elements that are located along the line we created here. So as you can see, we have some silicon and some sodium and sulfur and lead and gold, and this is all really good right here because this is what we're expecting to see in our sample. And finally, a last method of analysis we're going to look at is called elemental mapping. And we use this to get a more detailed reading of where certain elements are located, as well as observing their relative intensities across a larger area. So, to do this, we're going to click on Mapping. And from here, we can drag this little box to make it as big or small as we want, and cover the dimensions. And we can drag this so we can get a little elemental map. I'll come down here and place it over this spot again. And then we can hit Collect. And same as before, we got to confirm our elements. So we hit OK once again. And then it will start creating our map. And there's a couple of really cool things here. So we can look at a map of each element that's present. So as we can see here, we can look at a map of just silicon and where we're observing that. And that's all this purple here. Or we can look at the sulfur. I'll unclick that. Same with the lead and the gold here. And another really cool thing we can do is that we can hit overlay on the image. So this allows us to look at the map and our actual image all at once. As you can see here, we can also look at these numbers here and kind of compare the intensity. So as you can see here, we have a lot of silicon. And this is expected because our entire substrate is a silicon substrate. So we expect this to be very intense in our readings. While down here we have sulfur and lead and gold, which will have similar intensities because those are the components that are bonding. So when looking at the intensities here, we can do some of the analysis that we do every day with this. As you can see, our lead here is less intense than our sulfur and our gold. And this makes sense when considering how everything is bonded, and also the fact that our substrate is coated in lead, and we also have gold nanoparticles on that. So it makes sense that of these three, that we have the most gold here. And then finally, when doing this kind of analysis, we can look at the file in a Word document that it creates for us. So we can come up here. This is really helpful to come back to our data later, because sometimes we might not be looking for a specific thing in the moment, but we think of something later that we think would be important to check back at our samples and look at what we have. So all in all, this is a really important feature of the SEM, and we hope that you all get the chance to work with us on it later.
To enhance students' research and education experience, we will offer a special topic course this coming May semester. Physics 1980, Imaging the Invisible World. Midterm, like 3 weeks, from May 10th to 28th. The lectures will be on Monday, Tuesday, Wednesday, Thursday, Friday, from 8 to 11. And there are 2 different lab sessions, one on Tuesday, Thursday, another one on Wednesday, Monday, from noon to 3 p.m. And this is a 4-credit course designated with Hamline Plan Natural Science. And this course will teach students fundamental scientific theories behind optical instrument, including light microscope, scanning electron microscope, transmission electron microscope, NJ dispersive X-ray spectrometer, and how to use the instrument to investigate scientific questions, analyze and interpret data, communicate and share scientific findings with community through creative technical writing and microphotography. We give some examples, using different detectors you can observe different features. You also different resolution. You can to show more details of the samples or different materials. So use this instrument carefully to investigate scientific questions. Otherwise, sometimes you may make false statement. And also, how to share with community, because sometimes the reader, they don't know electron microscope. You can take tons of images, but you need to pick up or select representative image to share your scientific findings with community in the layman term. And each student will conduct an individual research project. They will take SEM images and select some of them to edit and submit for the Microscope Today Micrograph Awards competition. So through this course, the student will learn how to utilize the ESAM and Hamline for the research project, education, and community service. This is combine both science and arts. Instead of using a camera or cell phone to take pictures, this one we use scanning electron microscope to take image and to share with community. So that is, we encourage like students in the multidisciplinary in arts, anthropology, biology, biochemistry, chemistry, environmental studies, environmental science, forensic science, material science, physics. I just name those, but you can use this one for your study at Hamline. Thanks for attending this webinar. Please provide feedback and your comments through this website, basically for question survey. Right now, we are going to take questions. Meanwhile, after this webinar, if you have more questions, please send email to me. Thank you.